Class 12, Chapter 4, Principle of Inheritance and Variation. Here we will discuss four topics: genetics of Mendel's law of inheritance, inheritance of one gene, inheritance of two genes, sex determination, mutation, and genetic disorder. Okay, now let's start. Have you ever wondered why an elephant always gives birth only to a baby elephant, not some other animal, or why a mango seed forms only a mango plant, not any other plant? So let's discuss about it. Given that they are the offspring identical to their parents, or do they show difference in some of their characteristics? Have you ever wondered why these siblings something looks to so similar to each other, or something even so different? So these and several related questions are dealt with scientifically in branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parents to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Human knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breed examples Sahiwal cows in Punjab. We must, however, recognize that through our ancestors knew about the inheritance of characters and variations. They had very little idea about the scientific basis of this phenomena. Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiment on garden pea for 7 years, 1856 to 1863, and proposed the law of inheritance in living organism. During Mendel's investigation into inheritance patterns, it was for the first time that aesthetically analysis and mathematical logic were applied to problem in biology. His experiments had a large sampling size which gave greater creativity to the data and he collected. Also, the confirmation of his inference from experiments on successive generations of his test plants proved that his result pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite traits, as tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inherent in them. Mendel conducted such artificial pollination cross-pollination experiments using several true breeding pea lines, a true breeding lines in one that having undergone continuous self-pollination shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seed, yellow or green seed, inflated or constricted green or yellow pods, or tall or dwarf plants. Inheritance of one gene. Let us take an example of one such hybridization experiment carried out by Mendel where he crossed tall and dwarf pea plant to study the inheritance of one gene. He collected the seed produced as a result of this cross and grew them to the generated plants to the first hybrid generation. This generation is also called the filial progeny or F1. Mendel observed that all the F1 progeny plants were tall like one of its parents, none were dwarf. He made similar observation for the other pairs of traits. He found that F1 always resembled either one of the parents and that the trait of the other parents was not seen in them. Mendel then self-pollinated the tall plant and to his surprise found in the filial 2F generation some of the offsprings were dwarf. The characters that was not seen in the F1 generation was now expressed. The proportion of the plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and the dwarf traits were identical to their parental type and did not show any blending. That is all. The offsprings were either tall or dwarf, none were in between height. Similar results were obtained with other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on this observation, Mendel proposed that something was being stably passed down unchanged from parents to offspring through the gametes. Over successive generations, he called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles, that is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of the height, T is used for the tall trait and T for the dwarf, and T that and T are the alleles for each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T, small t, or small t, small t. Mendel also proposed that in the true breeding tall or dwarf pea variety, the allelic pairs of genes for the height are identical to homozygous capital T, and small t respectively, and are called the genotype. And plants, while the descriptive terms tall or dwarf are the phenotype, then what then would be the phenotype of the plants that had a genotype capital T, small t? And as Mendel found the phenotype of the F1 heterozygote capital T, small t to be exactly like the capital T parents in appearance. He proposed that in pair of dissimilar factors, one dominates the other and hence called the dominant factor, while other called the recessive. In this case, capital T tallness is dominant over the small t for dwarfness, that is recessive. He observed identical behavior for all other characters traits pairs that he studied. It is convenient to use capital and lowercase and an alphabetical symbol to remember this concept of dominance and recessiveness. Uh, do not use capital T tall and D for dwarf, because you will find it difficult to remember whether capital T or D are alleles of the same genes characters or not. Alleles can be similar as in the case of homozygotes, that capital T small t, and dissimilar in the case of the heterozygous capital T small t. Since the capital T small t plant in heterozygous for the genes controlling one character, it is monohybrid, and the crossbreeding capital T small t is a monohybrid cross. From the observation that the recessive parents and traits is expressed without any blending in F2 generation, we can infer that when the tall and the dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other, and only one allele is transmitted to a gamete. The segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossing. In this way, the gametes of the tall capital T plants have the alleles capital T, and the gametes of dwarf small t have the allele small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and small t from the other parents, then through the egg, are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrids that have capital T small t, since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, and F1 and F2 plants can be understood from a diagram called Punnett square, as shown in figure. It was developed by British genetics Reginald C. Punnett. It is graphical representation to calculate the probability of the all possible genotypes to offsprings in a genetic cross. The possible gametes are written on the two sides, usually the top row and the left columns. All possible combinations are represented in the boxes below in the square, which generates a square output form. The Punnett square shows that the parental capital T male and dwarfed small t female plants, the gamete produced by them, and F1 T progeny and F1 plants show genotype capital T small t are self-pollinated. The symbol male and female are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the phenotypic when saturated a typical monohybrid proportion when fertilization takes place, the pollen grains of the between true breeding tall plants T have a 50% chance of pollinated eggs of the genotype T, as well as the of genotype small t. Also, pollen grain of genotype T have a 50% chance of pollinating eggs of a genotype small capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be genotype TT. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to TT and half lead to capital T small t and 1/4 to small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, third fourth of the plant are tall when some of them are tall, while others are tall. Externally, it is not possible to distinguish between the plants with the genotypes tall and tall. Hence, the within the genotypic pair tall, only one character tall is expressed, and the character of capital T tall is said dominant over the other allele, that is dwarf character. It is thus the dominance of the one character over the other that all the F1 are the tall, though the genome is in the F2, third fourth of the plants are tall, and genotypically half are capital T small t and only 1/4 of the are TT, and this leads to the phenotypic ratio third fourth tall and 1/4 tall and 1/4 small. Dwarf, that is 3:1 ratio, but a genotypic ratio 1:2:1, and 1/4 is to 1/2 is to 1/4 ratio mathematically considerable to form the binomial expression ax plus by whole square, that is the gametes bearing genes capital T or in small t in equal frequency of half. The expression is expanded as below given. Mendel self-pollinated the F2 plants, found the dwarf F2 plants continue to generate dwarf plants in F3 and F4 generation. He concluded that the genotype of the dwarf was homozygous. What do you think he would give had self-pollinated at F2 plant? From the preceding paragraph, as it clear that though the genotypic ratio can be calculated using a mathematical probability by simply looking at the phenotype of a dominant trend, it is not possible to know that the genotypic composition, that is, for example, whether a tall plant or F1 or F2 has decomposition cannot be predicted. Therefore, to determine the genotypic of all plants at F2, Mendel crossed the tall plant from the F2 plant with a dwarf plant. This is called test cross. In a typical test cross, an organism showing a dominant phenotypic and is crossed with a recessive plant instead of self-crossing. The progeny of such a cross can easily be analyzed to predict the genotype of the test organism. Figures show the results of the typical test cross where violet color is dominant over the white color. Based on the observation of the monohybrid cross, Mendel proposed two general rules of consolidate his understanding of inheritance in the monohybrid crosses. Today, the rules are called principle or law of inheritance, and the first law of dominance and the second law of segregation. Law of dominance. Characters are controlled by the discrete unit called factors. Factors occur in pair. In a dissimilar pair of factors, one member of the pair dominates the other. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in F1 and the expression of the both in the F2. It explains the proportion 3:1 obtained at the F2. Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation. Though one of these is not seen at the F1 stage, though the parents contain two alleles during gamete formation, the factors or the alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while the heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Incomplete dominance. When an experiment or a P were repeated using other traits in other plants, it was found that the something and the F1 had a phenotypic that did not resemble either one of the parents was been in between the two. The inheritance of the flower color in the dog flower is a good example to understand incomplete dominance in a crossbreeding true breeding red flower and true breeding white flower, and the F1 was pink when the F1 was self-pollinated with F2, resulting in the following ratio 1:2:1. Here the genotypic ratio were exactly as we expected in any Mendelian monohybrid cross, but the phenotypic ratio had changed from 3:1 dominant is to recessive ratio. What happens was the capital R was not completely dominant over the small r, and this is possible to distinguish pink from the red and white. Explanation of the concept of dominance. What exactly is dominance? What are some alleles dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait in a diploid organism. There are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not only always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of the gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal or less enzymatic enzyme, or a non-functional enzyme, or a no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotypic trait that is result in the transformation of the substrate. Such equivalent alleles pair are very common, but if the alleles produce a non-functional enzyme or no enzyme, the phenotypic may be affected. The phenotype trait will only be dependent on the functioning of the modified allele. The unmodified allele, which represent the original phenotype, is the dominant allele, and modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because of non-enzyme is produced. Codominance. Till now, we were discussing cross, where the F1 resembles either of the two parents or was in between, but in case of codominance, F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human being. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells have sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene has three alleles, IA, IB, and IE. The alleles IA, IB produce a slightly different form of the sugar, while the allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I genes are completely dominant over small I. In other words, where IA and I are present, IA express because does not produce any sugar, and when IB and IB express, IA and IB are present together, they express their own types of sugar. This is because of codominance. Hence, red blood cells have both A and B types of sugar. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore a total 6 different genotype of the human ABO types. How many phenotypes are possible? Calculated now. Realize that there is an example of ABO blood grouping also provides a good example of multiple alleles. Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in the individual only the 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seed is controlled by one gene. It has 2 alleles. Starch is synthesized effectively by homozygotes, and therefore large starch grain are produced. In contrast, homozygotes have lesser efficiency in starch synthesis and produce a smaller starch grain. After maturation of these seeds, capital B seeds are round and small b are wrinkled. Heterozygotes produce round seeds and seems to be dominant allele, but the starch grain produced are the intermediate size seeds. So if the starch grain size is considered as the phenotype, then from the angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of the gene or the product that it has information for. It depends as much on the gene's product and the production of a particular phenotype from this product, and it does not have a particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. Inheritance of the two genes. Mendel's also worked the cross pea plant that were differed in two characters, as seen in the cross pea plant that has seed with the yellow or color or round shape, one that has seed of green color and wrinkle shape. Mendel found that seed resulting from the crossing of the parents had yellow colored and round shaped seeds. Here you can tell which question. Thus, yellow color was dominant over green and round shape dominant are over wrinkle. These results were identical to those he got when he made separate monohybrid cross between yellow and green seed plants and between the rounds and wrinkle seed plant. Let us the genotypic symbol Y for dominant yellow seed color and small y for recessive green seed color. R for round shape and small for single wrinkle seed shape. The genotypic parents can be written as the following, and the cross between the two plants can be written down as in the figure. Parents plant the gametes unit of fertilizer to produce F1 hybrid. When the Mendel self hybridized the F1 plants, found that third-fourth of the F2 plants had yellow seed and one-fourth of the green. The yellow and green are segregated in the ratio 3:1 ratio. Round and wrinkle seed shaped also segregate in 3:1 ratio, just like a monohybrid cross. Law of independent assortment. In the dihybrid cross, the phenotypic round yellow, yellow green and wrinkle green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green, 3 round and 1 wrinkle. This derivation can be written as follows. Based upon such observation on dihybrid cross, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two parents of the traits are combined in a hybrid, segregation of one pair of characters is independent of the other pairs of the character. The Punnett square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of eggs and pollen in the F1 plant. Consider the segregation of one pair of genes R and R. 50% of the gametes have gene R and the 50% have a small r. Now, besides the gametes having a small r, capital R should be having the allele capital Y or a small y. The important things to remember here is the segregation of 50% as capital R, 50% small r independent, and segregation 50% capital Y and 50% small y. So, thus there are 4 types of gametes. 4 types of R, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Each frequency 25%, 1/4 of the total gamete produced. When you write down the 4 types of eggs, pollen on the sides of the Punnett square, it is very easy to derive composition of the zygotes that give rise to the F2 plant. Although there are 16 squares, how many different types of genotypics, phenotypics form? Chromosomal theory of inheritance. Mendel published his works Inheritance of Characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy in those days and his work could not be widely publicized. Secondly, his concept of genes or factors in Mendel's established discrete unit that control the expression of the traits and of the pair of the alleles which do not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variations seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that the factors were discrete units, he could not provide any physical proof for the existence of factors or said what they were made of. In 1900, 3 scientists, De Vries, Correns and von Tesmark, independently rediscovered Mendel's result of the inheritance of the characters. Also, by the time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, colored bodies, as they were visualized by staining. By 1902, the chromosomes movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of the chromosomes was parallel to the behavior of genes and used chromosomes movement. To explain Mendel's law, recall that you have studied the behaviors of the chromosomes during mitosis and during meiosis. The reduction division, the important things to remember that are chromosomes as well as the genes occur in pairs. 2 alleles of the genes are pair located on the homologous site of the homologous chromosome. Now, Sutton and Boveri argued that the pairing of segregation of a pair of chromosomes would lead to the segregation of pair of the factors they carried. Sutton united that the knowledge of chromosomal segregation with Mendelian principles called the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis of the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simply synthetic medium in the laboratory. They complete their life cycle in about 2 weeks. A single mating could produce a large number of progeny flies. Also, there was a clear differentiation. One of the sexes, the male and female flies, are easily distinguishable. Also, it has many types of hereditary variation. This can be seen with low power microscopes. Linkage and recombination. Morgans carry out several dihybrid crosses in Drosophila to study genes that were sex-linked, and the crosses were similar to dihybrid cross carried out by Mendel in P. For example, Morgans hybridized yellow bodies, white-eyed females, or brown bodies, red-eye-colored males, and intercrossed their F1 progeny. He observed that two genes did not segregate independently of each other, and F2 ratio deviated very significantly from the ratio 9:3:1. Morgan and his group knew that the genes were located on the X chromosomes. One saw quickly that when the two genes in a dihybrid cross were situated on the same chromosomes, the proportion of the parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes, and coined the term linkage to describe the physical association of genes on a chromosome, and the term recombination to the describe generation of non-parental gene combination. Morgan and his group also found that even when the genes were grouped on the same chromosomes, some genes were very tightly linked, and while others were loosely linked. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% of the recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used the frequency of the recombination between the genes pair of the same chromosomes as a measure of the distance between genes and mapped their position on the chromosomes. Today, genetic maps are extensively used as a starting point in the sequencing of the whole genomes was done in case of the Human Genome Sequence project described later. Polygenic inheritance. Mendel's study described the traits that have distinct alternate forms such as flowers colors with their either purple or white, but if you looked around you will find that there are so many traits which are not distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as distinct in alternative, but a whole range of possible heights. Such traits are generally controlled by three or more genes and thus called polygenic traits. Besides the involvement of the multiple genes, polygenic inheritance also takes into account of the influence of the environment. Human skin color is another classic example for this. In a polygenic trait, the phenotypic reflects the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that the three genes ABC control a skin color in human, and with dominant form ABC responsible for the dark skin color and recessive forms small e ABC light skin color. The genotypic with all dominant alleles will have the darkest skin color, and that with all recessive alleles will have the lightest skin color. As expected, the genotypic with three dominant alleles and three recessive alleles will have the intermediate skin color. In this manner, the number of each type of alleles in the genotypic would determine the darkness or lightness of the skin in an individual. Pleiotropy. We have so far seen that the effect of a gene or a single phenotypic or a trait that there are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene. The underlying mechanism of the pleiotropy in most cases is the effect of a gene on a metabolic pathway which contribute towards different phenotypics. As an example of this is disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzymes phenylalanine hydroxylase. The manifest itself through the phenotypes expression characterized by the mental retardation and reduce the hair, skin and skin pigmentation. Sex determination. The mechanism of the sex determination has always been a puzzle before the genetics. The initial clue about the genetic chromosomal mechanism of the sex determination can be tracked back to the sum of the experiments carried out in the insect. In fact, the cytological observation made in a number of insects led to the development of the concept of the genetic chromosomal basis of sex determination. Hanking could trace a specific nuclear structure all through the spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to this structure as the X body, but he could not explain the significance. Further investigation by the other scientists led to the conclusion of the X body. Hanking was in fact chromosome, and that is why it given the name X chromosome. It was also observed that in the large number of insect, mechanism of sex determination in exotype, that is all eggs bear an additional X chromosome beside the other chromosomes. On the other hand, some of the sperm bear X chromosomes, whereas some do not. Eggs fertilized by sperm have X chromosomes because become female, and the pseudo fertilized by sperm that do not have X chromosome become males. Due to the involvement of the X chromosomes in the determination of sex, it was designated to the be sex determination, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of exotypes of sex determination in which the males have only one X chromosomes beside the autosomes, whereas the females have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination in a number of other insects and the animal mammals, including men. XY types of sex determination is seen where the both male and the females have the same number of chromosomes. Among the males, X chromosome is present with its counterpart distinctly small and called Y chromosomes. Females, however, have a pair of X chromosomes. Both the males and females bear same number of autosomes. Hence, the males have autosomes plus XY, while the females have autosomes plus XX. In human being and in Drosophila, the males have one X and the one Y chromosomes, while the females have a pair of X chromosomes besides autosomes. In the above description, beside you have studied about the types of sex determination mechanism, in that is exotype and XY type, but in the both cases X chromosomes, some gametes X chromosomes with some with Y chromosomes. Such types of sex determination mechanism designated to be example of male heterogamety. In some other organism, example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both the males and females, but two different types of gametes in terms of the sex chromosomes are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of females birds has to be designated to be the Z and W chromosomes. In these organisms, the females have one Z or one W chromosomes, whereas the males have a pair of Z chromosomes beside the autosomes. Sex determination in humans, it has already been mentioned that the sex determining mechanism in case of human is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in females, and whereas the presence of X and Y chromosomes are determinant of the male's characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry X chromosomes, and the rest of the 50% Y besides the autosomes. Females have, however, produced only one type of ovum with X chromosomes. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case of the ovum fertilization with the sperm carrying X, zygote develops into female, and fertilization of the ovum with Y chromosomes carrying sperms resulting into the male offspring. Thus, it is evident that it is the genetic makeup of the sperm that it determines the sex of the child, and it also evident that each pregnancy there is always 50% of probability of either a male or female. It is unfortunate that in society women are blamed for the giving birth for male children and having ostracized and ill treated because of this false notion. Sex determination in honeybee. The sex determination by honeybee is based on the number of set of the chromosomes in individual receivers. And offspring formed from the union of the sperm, and the egg develops as a female queen or worker. An unfertilized egg develops as a male means of parthenogenesis. This means that the males have half of the number of the chromosomes than that of a female. The females are diploid having 32 chromosomes, and males are haploid, that is having 16 chromosomes. This is called as a haplodiploid sex determination system and has a special characteristic features such as males produce sperm by mitosis that do not have father or thus cannot have sons, but have a grandfather can have a grandson. How is the mechanism different in birds? Mutation is a phenomenon which results in the alteration of the DNA sequence and consequently results in the changes in the genotype of the phenotype of an organism. In addition to the recombination, mutation is one phenomenon that leads to the variation in DNA. As you will learn in chapter 5, one DNA helix runs continuously from one end to other in such a chromatid in highly supercoiled form. Therefore, loss or gain segment of a DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes results in the abnormalities or aberrations Chromosomal aberration are commonly observed in cancer cells. In addition to the above, mutation also rise due to the change in the single base pair of DNA. This is also point mutation. A classical example of such mutation is sickle cell anemia. Deletion and insertion of base pairs and DNA cause flame shift mutation. The mechanism of mutation is beyond the scope and this discussion at this level. However, there are many chemical and physical factors that induce radiation mutation. There are referred to as mutagens. UV radiation can cause mutation in organism. It is a mutagen. Now, genetic disorder, a pedigree analysis. The idea disorder of the inherited has been prevailing in human society since long. This was based on the inheritability of certain characteristic features in families. After the rediscovery of Mendel's work of practice analyzing inheritance patterns of the trait of human beings began, since it is evident that the control crosses that can be performed in pea plant or some other organisms are not possible in case of human being. A study of female history about the inheritance of a particular trait provided an alternative. Such an analysis of trait in several of generation of a family is called pedigree analysis. In this pedigree analysis of the inheritance of a particular trait is represented in the family tree over generation. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbol used in the pedigree analysis have been shown in figure. As you have studied the chapter, you have every feature in any organism is controlled by one or the other gene located on the DNA represent in the chromosomes. DNA is a carrier of the genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes of alteration do not take place occasionally. Such an alteration of change in the genetic material is referred to as mutation, and a number of disorders of human beings have been found to be associated with the inheritance of the changed or altered gene or the chromosomes. Mendelian disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders or chromosomal disorders. Mendelian disorders are mainly determined by the alteration or mutation in the single gene. These disorders are transmitted to the offspring of the same lines, as we have studied in the principle of inheritance. The pattern of the inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common are prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in a question is a dominant or recessive. Similarly, the trait may also be linked to the sex determination, as in a case of hemophilia. It is evident that this excellent recessive trait shows the transmission from carrier female to male progeny. A representative pedigree is also seen in for dominant or recessive trait. Discussion with your teachers or design pedigree for characters linked to the both autosomes or sex chromosomes. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of the eye, resulting in failure to discriminate between the red and green color. This defect is due to mutation in certain genes present in the X chromosomes. It occurs in about 8% of the males and only about 0.4% of the females. This is because the genes that lead to the red-green color blindness are on the X chromosomes. Males have only on the one X chromosomes and females have two. The sons of a woman who carries the gene has 50% chance of being colorblind. The mother is not herself colorblindness because the gene is recessive. That means that that its effective is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease, which shows its transmission from unaffected carrier females to some of the male progeny, have been widely studied. In this chapter, a single protein that is a part of the cascade of the proteins involved in the clotting of the blood is affected due to this. In fact, affected individuals in a simple cut will result in the non-stop bleeding. The heterozygous females of the hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic. The family pedigree of Queen Victoria, as shown in the the number of hemophilic descendants, as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for the genes. This disease is controlled by a single pair of allele HBA, HBS. Out of 3 possible genotypes, only homozygous individuals for HBS show the disease phenotype. Heterozygous individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of the transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid bivalent at the 6th position of the beta globin chain of the hemoglobin molecules. The substitution of amino acid in the globin protein resulted due to the single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecules undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle cell-like structure. Phenylketonuria. This inborn error of the metabolism is also inherited as the autosomal recessive trait. The affect individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also excreted through urine because of its poor absorption by kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene. The defect could be due to either mutation or deletion, which ultimately results in the reduced rate of the synthesis of one of the globin chains that makes up hemoglobin. This causes of the formation of abnormal hemoglobin molecules, resulting in anemia, which is characteristic of the disease. Thalassemia can be classified according to which the chain of hemoglobin molecules are affected. In alpha thalassemia, production of alpha globin chain is affected, while the beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, HBA1, HBA2, on chromosome 16 of each parent, and it is observed due to mutation, or the chromosome 16 of each parent is observed due to mutation, deletion of one or more than 4 genes. The more genes affected, the less alpha globin molecules produced, while beta thalassemia is controlled by single gene HBB on a chromosome 11 of each parent and occurs due to the mutation of the one or both the genes. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is qualitative problems of the synthesizing an incorrectly functioning globin. Chromosomal disorder. The chromosomal disorder, on the other hand, are caused due to the absence or excess of abnormal arrangement of one or more chromosomes. Failure of segregation of chromatid during the cell division cell cycle results in the gain or loss of the chromosomes, called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosomes 21. Similarly, Turner syndrome results due to loss of X chromosomes in the human females. Failure of cytokinesis after telophase stage of the cell division results in an increase in the whole set of the chromosomes in an organism, and this phenomenon is called as polyploidy. This condition often seen in the plants. The total number of chromosomes in normal human cells is 46. Out of these, 22 pairs are autosomes and 1 pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosomes may be included in an individual, or an individual may lack one or any pair of chromosomes. These situations are known as the trisomy, monosomy of a chromosomes respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of the chromosomal disorders. Down syndrome, this cause of genetic disorder is the presence of the additional copy of the chromosome number 21. This disorder was first described by Langdon Down. The affected individual is short-statured, with a small round head, furrowed tongue, and partially open mouth. Palm is broad, characteristic palm creases. Physical, psychomotor, and mental development is retarded. Klinefelter syndrome, the genetic disorder, is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype of 47XXY. Such individuals have overall masculine development. However, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. Turner syndrome, such a disorder is caused due to the absence of one of the X chromosomes is 45 with XO. Such females are sterile with ovaries rudimentary, besides with the feature including lack of the other secondary sexual characters. So that's it. I hope you enjoyed. So thank you so much. Keep studying.